Today, we are going to discuss about the theory of Morgan. Morgan has proposed the theory of linkage, and he has conducted his experiments in Drosophila.
Before that, we have to discuss about the chromosomal theory of inheritance. This theory was proposed by Walter Sutton and Theodore Boveri in 1902. So they proposed the major points, that is, the chromosomes as well as the genes both occur in pairs. If you are taking human beings, 23 pairs of chromosomes, and each and every chromosome, the genes are in pairs. That means each gene, each character is controlled by a pair of genes. Then the second point, the genes are located on the same homologous chromosome. The two alleles of a gene pair are located on the same locus of a homologous chromosome. And along with that, you have to add each chromosome can carry many genes, and these genes are arranged linearly. That means one over the other. So one by one, all they are arranged linearly. Then at the time of gamete formation, third point, the pairing and separation of a pair of chromosome. When the chromosomes separate, along with that, the gene also will get separated. And out of one pair, only one factor or one gene only will move to the gamete. So they will separate at the time of gamete formation. And from that, only one gene which regulate a particular character will only move to that to the gamete along with the unpaired chromosome. These are the 3 points mainly proposed by the Sutton and Boveri and named as chromosomal theory of inheritance. So first one, chromosome as well as genes occur in pairs, and each chromosome consists of many genes and they are located in the chromosome and they are arranged linearly. At the time of gamete formation, the paired condition will segregate or separate, and out of that pair only one factor or gene or allele only will move along with the unpaired chromosome. Then later, this process this theory were elaborated with experimental basis with the help of T.H. Morgan, and he framed another term, that is linkage. T.H. Morgan, Thomas Hunt Morgan, he was doing his genetical experiments in Drosophila melanogaster, which common name is fruit fly. The specific features of the fruit fly, that is, it can be grown in a simple synthetic medium in the laboratory. Second point, they complete their life cycle in about 2 weeks. And single mating itself produce large number of flies. And they were showing contrasting characters. If they have red eye, one group they have means the other one will have white eye. Then very easily we can detect the sex, that is, here in the figure itself we can see male and female. Male is comparatively shorter and female is larger. Then they can be grown in a very cheap medium. That is either the vegetable waste, potato peel or banana peel in a small bottle. That is enough for them to culture. So very limited space and limited nutrient medium, that is cheap nutrient medium. Many generations within short time. One generation it will take only 2 weeks. So many generation within a short span of time it can be studied. Apart from that, this is the karyotype of the male. The male here, this one is the female. So you can see they carry only 4 pairs of chromosomes, and all the 4 it is of different shape. That is, here it is no centromere at all. Here at the tip, and here it is extreme in the center, and this one is somewhat submetacentric. So we have learned eccentric, acrocentric, telocentric, then submetacentric and metacentric. So all these 4 different types of chromosomes can be possible. So very easily we can distinguish their chromosome. Then the first pair is called as the sex chromosome. So sex chromosome in female carry 2 X chromosomes, and male carry 1 X chromosome and Y chromosome. The Y chromosome in Drosophila does not carry any genes. It is empty, and it is having the shape of a J shape. That means submetacentric type of chromosome. So J shape. So very easily we can detect their chromosome, the male and female also. So all these factors favored to find out or to helped him to come into the conclusions very easily. And Morgan, when he studied the dihybrid crosses, he found out that the ratios of test cross varies from what Mendel he said. Mendel he said a typical dihybrid cross, the result dihybrid test cross, the result will be always 1:1:1:1:1. That means all the different combinations will be available, can be resulted in equal proportion. When Mendel he conducted the same experiments by conducting 2 contrasting characters. One is for the body color of the Drosophila, and the other one is eye color of Drosophila. And he got the result slightly varying from Mendel. And from this, he proposed many generalizations. And he said, why this deviation? Why the number of recombinants, where recombinant means non-parental combinations, were less compared to Mendelian ratios, and parental combinations were more than the expected ratio. If the parent is having that particular combination, that exact combination were more in the test cross ratio. So from this he concluded that if the genes are located in the same chromosome, if the genes which regulate the expression. So Morgan he said why he got more parental combination in his result means because the selected characters were the genes which regulate that selected characters were located in the same chromosome. So he said if they are located in the same chromosome, they will have a tendency to inherit as it is to the next generation. That is why the parental combinations were more. And that tendency of the genes which are located in the same chromosome to inherit as it is to the next generation, he termed as linkage. The physical association of genes on the same chromosome. They will show more linkage. And he said one more term, there can be recombination. So why the non-parental combination is arising? If they have the linked tendency, why then the recombination? If it is completely going as it is, Then in the test cross ratio, they have to get only 1:1 ratio. That is the parental generation alone. But even then, his studies, the first cross in your textbook, that is given on the left side, the first cross, on the right side, the second cross. So here I am dealing only the cross A. That is lengthwise it was given. Now I have rearranged in a little bit different way to make it more convenient to understand. So here I am dealing with only the cross A, in which he took two characters. And this one is the parental generation. Don't see this side. You have to go only this one, detail. And here he has selected two characters. The first one, it is body color and the eye color. So he found out that these two colors are located in the X chromosome. So when we deal with X chromosome, the female Drosophila carry two X chromosome. That is why here it is given in this way, two yellow rod-like structures. So, and male will carry one X and Y, XY combination, one X and one Y. And Drosophila Y chromosome does not carry any gene. Okay, male Drosophila does not carry any gene, so it is empty. That is why J-shaped structure, somewhat like a J-shaped structure Y. And it is, so in this, the character, one allelic representation is enough for the male to express. One allelic representation is enough because Y will not have any paired condition. So male, the genes will be always X chromosome. The genes which are present only in X chromosome. So single character representation only, factor representation only will be there in male, whereas in female, two X chromosomes, so paired condition can be seen. Here it is unpaired condition, here it is paired condition. Now we will move on to his. And he said the body color and eye color is located only in the X chromosome. Only in the X chromosome. So female will have paired characters, paired factors. Male will have unpaired because Y is empty. So this factor, if you are clear, then first step will be over. And he crossed the female which carry yellow, white. These two traits are recessive. That is why we are representing with small y, small w, small y, small w. Both the characters, both the factors are recessive. That means it can express only in double recessive condition. So here there is no dominant allele, hence we are representing it as yellow, white. When we move on to the male, here brown body, because dominant, that's why we are representing small y plus. Dominant always recessive traits, small letter plus signal, plus symbol. So that is modern Morgan symbolization, Y plus. That means it is dominant. Then W plus means red eye. So brown bodied red eye. This is both these factors are dominant, hence we represent it as wild type. So even though when we say wild type, they represent brown body, red eye. So brown body, red eye, male. And he said if these two genes are located in the same chromosome, if these two alleles are located in the same chromosome, they will have a tendency to inherit as it is to the next generation. That means when they form gamete, the female, when they form gamete, one gamete will be, I'll take the pen. So here the one gamete will be, it's not coming out. One gamete will be, let me see whether, no, arrow mark only is coming. When one gamete will be of the same type, you see, that is if you are circling one X chromosome alone. So you will get one gamete which carries, since both of them are homozygous for one representation is enough. So one of this one will move when you are crossing it. You see, YW, one gamete, female gamete, and one gamete from the male gamete, that is Y plus W plus, another gamete. So these two gametic combinations, when it is appearing in the F1, since these two are dominant and these two are recessive, the offspring which is formed will be 2X chromosome, hence definitely they will be female. And on this X chromosome Y and W, and in this one Y plus and W plus. Since both of them are Y plus and W plus, the phenotypic trait will be brown bodied, red eye, or we can say wild type. So F1 offspring will be wild and it is female. The other possibility, this gamete suppose joins with the Y. So here Y is empty, hence the F1. The males will be having YW, that means yellow, white. So this is the F1 generation. Up to this, most of you will be familiar. Then the second part only in the textbook, little bit confusing way that I have presented in a linear arrangement. And this is F1 and F1 into F1. That means it is equivalent of test cross ratio. F1 is heterozygous dominant and the male partner is recessive. This is recessive. Yellow white is the recessive and brown red is the wild type. That means the dominant. So heterozygous dominant F1 with the recessive character. F1 double heterozygous condition with the recessive, because Y is empty, recessive character. As per the rule, we have to get 1:1:1:1:1. But he got entirely different in the F1 generation. How the results varying means these are the parental combinations. That is, here you see, if there is no crossing over, if there is, here it is the possibility, that is between the parents, between the female, the X and X chromosome, if there is no crossing over, you will get like this combinations. But instead of that, when you go here, the female, the same female only I have represented for the other possibility. If crossing over means you will get like this. If this itself, among this at the time of gamete formation, if it is crossing over means genetic exchange. The genes will exchange among themselves. So here I have marked like this way. This is the possibility of getting recombinant varieties. So here first we will move on to, so from here the Y can go this side and this Y plus can go this side. So if crossing over takes place, there will be another result. So first we will move on without crossing over, what happens. If there is no crossing over means this will act as one gamete, this is another gamete, and from the male this one gamete, another gamete will carry Y. So here now you see all the possibilities. This gamete, if fuses with this gamete, that is here it is Y, with the Y, X with Y. This X with this X means this one. So YW, again YW here. And this YW with the this one here only. This one means here you will get YW, YW. That means both will be yellow, white. One is male, the other one is female, because this gamete from the female, this gamete only, recessive character only fusing with these two. Then the next possibility, this gamete, that is Y plus and W plus, dominant character. Dominant character if fuses with the X chromosome of male. So 2X condition, you will get Y plus W plus as well as YW. this one. So this also, at least one dominant alleles are there, hence they are coming under the category of Y wild type, that is female. So 2X chromosome means it is female. Then this X chromosome combines with this Y chromosome. This X chromosome combines with this Y chromosome. You will get Y plus W plus and the Y chromosome. It is empty. So here the character expressed will be wild type. And here this possibility, this combination will get if there is no crossing over. And suppose if the crossing over takes place between these two X chromosome at the time of gamete formation. So that means they will cross over. These two genes will cross over. As a result, what will happen? This is the possibility I have represented in this without crossing over, like this gametic combination. If the crossing over takes place means the gametes can be of two different types. That is, here you see Y plus going with W plus. Y plus, that means this gene will shift to this side. This gene will shift to this side. And the next one, this one will move to this side. So you will see W plus W. If this one is going here means Y plus W. Then the next one you see Y and W plus. If this also going this side, so here Y, W plus. If any crossing is taking place, both simultaneously will not, any one. So you will get a recombined combination. So here the gamete will be varying from this. Here this is no crossing over, that is why as it is from the parent to the offspring. And from this as it is to the next generation. Here it is not like that. If they form in this stage, if they are a genetic exchange is taking place, the gametic combination of that gamete will be entirely different. If it is here YW means here it can be a recombined character. A bit of from this can move to this. That is why here two color, blue color as well as the yellow color, Y plus W. And another possibility YW plus. And like this recombined gamete have equal possibility of combining with the male. So if this gamete which is recombined or which is formed as a result of crossing over can cross with the male X chromosome. So you see YW, Y plus W, which is white with brown body. White with brown body, which was not there in this F1. White with brown body. Then the next one you see the same thing, this one. This one with again the X chromosome of male. So if the crossing over is taking place, then only I am saying this combination. You can see YW plus, that is YW plus, then YW. So here you see the new combination arises. Then again the next one with the Y, that is male gamete, Y gamete, X and Y. So this one will become male. This one also will become male. So these four sequence only they have given in the text, the down portion, the left side collectively. And this figure only on the right side it is given. So what is given in that left corner of that under the cross A, this one is on the left side, this one is on the bottom box right side. So if crossing over is taking place means these four results. No crossing over. If no crossing over means these four combinations can arise, which is exactly the parent combination. You see yellow white, then again yellow white. Then these two are wild type, which is also present in the wild type. But if crossing over takes place in this offspring, in this F1 female, then the combination is different, white with red eye, which is not there in the parental. then yellow with white eye, yellow with red eye, yellow with red eye. Yellow is recessive, red eye is dominant, which is also not present in the parental. Then the next one, it is white eye with brown body, male. It is not there. And then next one, it is yellow body with red eye, male. So that also not present in this one. So these four combinations are new. These four means actually white offsprings and yellow offsprings. White with red eye and yellow with red eye. No, white with brown body and yellow with red eye. White with brown body, it is not there. Yellow with red, that also not there. So these combinations arising, these combinations are called as non-parental combinations. This is caused due to recombination. Recombination as a result of crossing over. If crossing over is taking place between the gametes between the chromosomes, homologous chromosomes of the F1 female. Okay, if crossing over does not take place means exactly the parent combination, that is yellow white you are getting, wild type you are getting. But if exchange between the X chromosomes of the female, then the combination will be varying from the parental generation. So this is one possibility, this is another possibility. The first possibility, without crossing over, in your textbook it is in the extreme left side. And possibility of crossing over their gametic combination in that A part itself on the right side. So this I have made in this way to make it little more clear. And the same way he conducted, he didn't stop with one cross itself, he conducted another cross B. In that combination of characters only different. In the cross B, two characters, that is the eye color, not eye color, eye color and which one? The wing size. And in that he found out the percentage of crossing over the non-parental combinations increased. And then he come into the conclusion, whenever the percentage recombinants are increasing, that means the distance between the genes are more. If more the distance, more chances of genetical exchange, the crossing over. Because here, if it is located here, means very easily it can break into pieces. Then it can rejoin. So when it is more chances of recombination, then the non-parental combinations also will keep on increasing. So from this he concluded that the strength of linkage between the genes which are located in the homologous chromosome or within a chromosome depends upon the crossing over. Okay, so the linkage tendency is depending upon the distance between the genes. If the genes are very close, then linkage will be very high. If the genes are very wide apart, then the linkage will be very weak. So here in the first cross A, where the body color and eye color is taken, there the genes were very close. When the wing size and the body color, I think so, body color is taken, the distance were very apart. Not in this figure, I haven't included that figure. There the distance were wide apart. And as a result, there the parental combinations were approximately 63 percentage, and the non-parental recombinants were approximately 38 point something. That indicates the result of that one is the distance between the genes which is located on a chromosome will regulate or will directly related to the percentage of recombinants. Okay, percentage of recombinants will increase whenever the distance between the genes are more. And recombinant frequency will be high when the linkage is weak. Recombinant frequency will be low when the linkage is, strength of linkage is very high. So from this he has concluded. Now we'll move on to the next topic, that is one of his student, that is Sturtevant, Alfred Sturtevant. He used this recombinant frequency. So what do you mean by recombinant frequency? The number of recombinants which is formed in a test cross divided by total number of offsprings in a test cross. Suppose in a the 100 offsprings were produced in test cross. Out of that 20 recombinants appeared, means 20 divided by 100 into 100, that is percentage. So we will get 20 by 100, 100 will be equal to 20. So that means the two characters, the distance, two genes what he selected, the distance between that will be 20 units. In order to honor his master, because his team head was Morgan, his teacher. So in order to honor his teacher, he named that unit of distance between the gene unit, he named it as 1 Morgan unit. If 1% of recombinant frequency means the distance between the gene 1 and 2 is 1 map unit or 1 Morgan unit. Then if it is 2% means 2 unit, 30% means 30 map unit. So he used the position, or he used recombinant frequency for identifying, for measuring the distance between the genes, and that helped him to map the position of genes on the chromosome. That helped him to map the genes on the chromosome. So in this way, so when we are representing, so suppose gene A, then gene B, and gene C. So A and B, the combination, the gene is 2 unit, and A and C is 4 units. A and C is 4 units. So you can see A and B, then B and C. And A and C, if combination you are taking, the distance is 4, means you can say the order of gene is A, B and C. In that way, he developed a technique to apply his information, or the information of frequency of recombinants, to position the genes on the chromosome. So that was the first chromosomal map. So first chromosome map was invented by, or discovered by, or invented by the Alfred Sturtevant. Then this information he passed on, or that itself has given as the starting point for sequencing the whole genome. And more or less based on this principle only, we have developed the human genome sequencing project, human genome project later. That is with the help of, instead of manually locating the position of gene, we were doing it the same procedure with the help of software, computer made software and hardware. But the first chromosome map was constructed by Alfred Sturtevant on Drosophila. Okay, now we'll move on to this sex determination. So this is very simple topic only, just I am going through 2 types of sex determination, because sex chromosome, the chromosome itself broadly classified into 2 categories, autosome and sex chromosome. Autosome will control the somatic characteristics, sex chromosome will control the genetic characteristics and the reproductive characteristics. So there are 2 types of sex determining mechanism, that is male heterogamety and female heterogamety. So male heterogamety means male members will produce 2 types of gametes, And this gametes itself can be, we follow broadly, it is XX XY chromosome and XX XO condition. So that means male members will have different types. That is, one is X, the other one is Y. And the females will be homogametic. They produce the same type of gametes in female, and whereas in male they produce two types of gametes. X bearing gametes 50%, Y bearing gametes. The typical example is human being. The same way in grasshopper, XX XO. The females will produce the same type of gamete, that is X bearing gamete. Male will produce 50% X bearing and 50% without X, XO condition. That means in their whole genotype, one chromosome will be less. Then here female heterogamety means ZO ZZ. That means ZZ ZO or ZZ ZO ZZ. Here the male is homogametic. Male is homogametic and female is heterogametic. Then ZW ZZ, fowl. That is the birds and all. The male is homogametic, Z bearing, and two types of eggs they can produce. Either they can bear Z chromosome or they can bear W chromosome. Apart from that, there is one more, that is haplodiploidy. Honeybees, they are following a different type of sex determination. This is the diagram what is given in the textbook, sex determination. And this X body, they found out, that is X body, it is Henkin scientist only, they found out a darkened body in the female fly, the egg of the female. So he said there is a darkened body and he named it as X body. Then later only he found out that X body is the sex chromosome. And in the sperm, is the sperm or the egg responsible for sex of the chick? So it is in the chick, it is female heterogamety. The females are producing two types of gametes. One is Z, the other one is W. So the female egg determine what type of sex is produced in the next generation. And human beings and Drosophila, XX XY condition. This is just a diagrammatic sketch of sex determination. Here it is male heterogamety, and this one also male heterogamety. That is, one is XX XY means here in the grasshopper XO XX. Okay, XO XX. So here the male 44 autosomes and XY, female 44 autosomes and XX. When the male forms, two types of gametes they are bearing, the sperms with egg along with the 22 autosome and the sperm with Y with 22 autosomes. So when you are representing then from the textbook, this representation is good. And females will produce 22 autosome plus X, 22 autosome plus X, same type of gamete. So it is females are homogametic in this, males are heterogametic. So the different combination in that way, which one is determining the male, the type of sperm determine the sex of the offsprings. More or less the same way, only here in grasshopper, the male are heterogametic, female are homogametic. So autosome plus X bearing sperms, autosome plus without X bearing. Then here female, the same autosome plus X bearings. Ova will be produced. So if this gametic combination, it will give rise to female. If this gametic combination, it will give rise to male. This one is a typical example for female heterogamety. The female fowl will produce, hen will produce two types of gametes. Either it can be Z bearing gamete or it can be W bearing gamete. Two types of ova, autosome plus W, autosome plus Z. Then males are homogametic, the same type of gamete, that is A autosome plus Z bearing. When we cross it, you can find out that it is actually the sex is determined by the female. Now we'll move on to the sex determination in honeybee. It is haplodiploidy. So here it is female and male. And here female always diploid and male always haploid. 32 chromosome here, here it is 16 chromosome. And the gametic formation in female, it is by meiosis, reduction division. So the gamete will receive only 16 chromosomes. And in male, it is mitosis. That means it is equational division. N will carry 16 chromosome. And here, if the egg happen to directly develop into, without fertilization, if an egg from the female directly develop into offspring, then the haploid male will be produced. So without fertilization, if they develop, that process is called as parthenogenesis. Without gametic fusion, that means the egg directly develops into an offspring that will turn into male. Instead of that, if male gamete, that is this one, fuses with the male egg, female, that is egg, the gametic fusion, sperm egg combines with sperm means fertilization takes place. That will develop into a female. So fertilized egg develops into female, unfertilized egg develops into male. The process behind is parthenogenesis. Here the process is fertilization. So here it is haploid and here it is diploid. Hence that type of sex determination is called as haplodiploidy. This diagram also copy into your notebook. With the help of this you can explain it. So with this I am concluding the revision part. Thank you very much.